Okay, yeah, all right. Again, thanks for organizing and get me invited here. It's an amazing place. I'm Lin Su. I'm a new lecturer in Queen Mary, so I just started my lab at this fancy building a few months ago. So it's in London, so if anyone is close by, just let me know. I'm happy to invite you to go around these buildings. We even have some plants, greenhouse on the top of that, and you can see the landscape of London very nicely. So let's go back to the topic today. So I assume I myself will be the beyond of the scope of this workshop, but the idea is trying to figure out if we can have some connections and possibilities to collaborate together. So I work with biohybrid systems, and I have to say thanks to Josh, so I don't need to introduce the extracellular electron transfer anymore, but we work all with the bacteria that can do electron transfer across the membranes and sending signals. And usually it's both ways. So the bacteria can sending signals to the outside, for example, an electrode, or they also can taking electrons from electrodes inside the bacteria and power some reactions. So if you talk about application-wise, we can do energy-based transfer. So there's microbial fuel cells. You can just connect the bacteria and power a cell phone. And you can do water treatment, like all the hazardous things in the water stream. You can use this technology to naturalize it. And you can do electrosynthesis, so that's the reverse electron transfer. You have some clean energy or clean electricity, you power the bacteria to do chemistry. And of course, as Josh nicely introduced about sensing, so we can also take the electron transfer as a matter of information transfer. So we can do biosensing, hopefully we can do some bioelectronics with some very complex design, or biocomputing, or even we can do electrogenetics. So basically the idea is you have electro signal to control the gene expression, then further control the behavior of your engineered bacteria. I have to explore the electron transfer a little bit for the concept of the projects I'm going to mention. So we talk about electron transfer for the pure electrons through the different machinery and proteins, but nowadays we also combine small molecules, which could be a source of the electrons and delivered to the bacteria, which by means we can have hydrogen, CO, or formate, all these small molecules. Again, we're trying to use the concept of small electron carriers. So again, my group is just newly started, and there's multiple directions I've been working in the past, and we want to carry on. So I study like different electron transfer pathways in different organisms. We do biosensing, so Josh is already introduced to that part. We do engineering, both the bacterial side and material side, trying to enhance the electron transfer. And last not least is what I've been done through my postdoc in Cambridge to do some solar conversion connected with the electron transfer for the bacteria. So today, I think I will just focus on some examples on the last application or the research. And again, trying to give some ideas and to see if there's some synergies from the audience and we can do with the PFAS. Again, this is the concept of the research we did. So you have light converting to the reducing the CO2 and to the CO, and then feed to the bacteria, and you have products. And again, this is the adaptation what we did. So this gas fermentating bacteria, it can use sugar. So fructose is their favorite food to survive. And in the meantime, they can also change their metabolic pathway a little bit and to use the gas. Well, we figured out in our project is actually it's known in the community of this gas fermentating bacteria. Is not publicly announced that. So when you do this kind of experiment, you need to adapt them a little bit. So you need to always give them sugar and same time give them syngas and slowly remove the sugar. Then bacteria will just fully rely on the syngas. I think it's some metabolic pathway they need to kick off. So what we did is we find out, okay, so instead of doing 1 or 2 rounds of this adaptation, let's do a longer term adaptation. So those bacteria, they growing surprisingly slow. So usually you need to take weeks for them to grow. So each of the transfer take weeks. So it's not one generation. I think each of the transfer has multiple generations for bacteria developing. And then we benchmarked the outcomes. So it depends on how you grow the bacteria with the gas. So we have to batch state. So basically you have a sealed bottle, you give them gas sitting there and they grow, or you have a flow setup, you keep giving them the gas and see how they grow. So it's really the concept to prove we achieve some successful in the adaptation. So the red-ish color shows still like the OD increase get increased, and also the products of acetate and ethanol also get boosted through the different types of the growth. I think especially looks way better in the flow batch, so you can reach almost 100 millimolars of acetate by the end of 1 week incubation. So I was in a chemistry group, so of course we want to be very careful about chemistry happening, and we need to prove that the acetate and ethanol product is really coming from the gas, not from some bio waste or the biomass or whatever the rich media bacteria has accumulated stored inside. So we did C13 labeling, and you can find. Although it's not 100% of the carbon we fixed from the C13 gas, but dominantly you can see all the split peaks, they are either 1 or 2 carbons labeled from the C13. And we also did some more complex experiment trying to figure out where is the limited step during the adaptation. Maybe I'm not going to the details, but basically we fit them also formate, which is the meat product when they're doing the gas fermentation, and then you can see they also have different rate of taking the isotopic label formate and generating isotopic labeled acetate. So those are all good and solid that we have adapted string and they behave better, and that's the step of our prototype or concept is really putting things together to do. We call it a cascade reaction or domino reaction or biohybrid reaction, depends how you want to get people excited. But again, generally what we do is you have a flow system with the gas pipeline. So the gas fill into a photoreactor chamber, converting the gas and then feed into the bacteria. And again, those are the final presents we have in the prototype, comparing the two strings. The wild type string without adaptation, they barely grow during the process, and the accumulation of the acetate as a product also is, you can barely to say they have done anything with the converting the gas, but with adapter one, you can see obviously compared to time 0, you have accumulated acetate and bacterial growth. So this is basically so far what we achieved. I'm going to change it a little bit, go back to the biohybrids, because people can easily just challenge me. Okay, what do you have to do? You just have 2 chambers and link the with a gas pipeline. You cannot call it a biohybrid system. So what actually the other efforts we have trying to do is how we make a real biohybrid system by combining some click chemistry there. So I'm still in the always still in the concept of how we making this biohybrid system to using solar energy. So this is a diagram like nowadays how people doing this kind of research. You have something photosensitizer can absorb the solar, and then you have something that can do the chemistry, and you put them together. In terms of the waste, you can either just mix them together, or you can have some small molecules or link some things together. One of the good demonstration in the field before our publication is on the corner. So there's some ruthenium dye and use some sulfur bind to link or covalent attached to a redox protein to do the electron transfer. And then later, our group or Erwin's group back in Cambridge think about ruthenium dye is kind of not sustainable photosensitizer. Can we do a more green photosensitizer in terms of the carbon dots we're using there? And we optimize the click chemistry to make the carbon dots as the photosensitizer and linked to redox protein. So the MTRC, hopefully you still remember what Josh mentioned, the complex that bacteria using to deliver electrons. So MTRC is the most outer membranes that can do the redox. of course, eventually the idea is to put into whole bacteria. But for now, what we did is, so this shows the wild type side group, and in the 657 side is a tyrosine residue, and what we did is we mutated into a cysteine. So you have the sulfur there, then later you can do the click chemistry to link with the carbon dots. So apologize, I'm not a chemist or not a real chemist, so I'm not going to go to the details how we managed to do that. So collaborators done that work. Basically, what we do is we have a malonide group functionize the carbon dots first, and then linked to the cysteine to have the click chemistry to bind together. So that's the hybrids we have been making. And of course, to get this thing to do the downstream reaction, you need to go through the columns and to purify them, collecting them. But as biologists may be familiar to look at the gels, so what we did is if you first add the third sample, so that's the real linked one. So we have the mutated MTRC and have the melamide function carbon dots. When you link together, you can see from the drop position, maybe it's not that obvious or convincing, like the band is slowly lower than the average of the other samples. So that's the hint that that protein gets heavier and has a carbon dots bind to them. And of course, there's issues like this is a native page gel, so the surrounded electric field also changes when you have a carbon dots. So the movement is a little bit tricky. So that's why we also did UV illumination. So protein itself without any staining, without any dyes, it shouldn't be fluorescence during this condition. But because the carbon dots is a nice absorber and also emit light wavelengths after the UV, so you can see a very light, well, it's not actually a light, but you can see the fluorescence of the carbon dots when it binds to the protein, so you see the band. The otherwise, if it's not band, so for example, line 2 or line 5, you have the smear of the carbon dots. So it's really a hint that we managed to do this hybrid carbon dots and the protein thing. And of course, we do experiments to prove. So this is a UV-based spectra. So when you shine light, activated carbon dots, and carbon dots pass the electron to the cytochrome or to the MTRC, it will be reduced. So you have the heme peak from 410 shifted to 420. So it's easy for you to detect how reduced you can do, and you can see our hybrid system, they will be being reduced way faster than the wild type ones. So that's evidence that we have been make that working. So those are really the two small stories. Again, the idea is trying to say if there's anything we can do together to contribute to the bigger goal to get rid of the chemicals we don't want. Really, I want to thank all the collaborators to support the work. So most of the work have been done in Aaron's lab back in Cambridge, and we also have nice collaborators in Cambridge, and also collaborators like Julia and Jessica from UEA. So they really helps a lot with the protein purification. And then we have Lars, he was in Leeds University, but now moved to Netherlands. And he also contributed a lot with the click chemistry. And of course, thanks to the funding to support the project get move on. Of course, thank you for listening, and happy to take any questions.